All right, this is part 3 of 4, with 4 parts of the notes to cover. Hopefully you went back and did your job there. All right. Checkbox on notes. Make sure you're aware of that. So we're talking about the law of independent assortment now. This is the third of the 3 laws that Gregor Mendel came up with. And this states that different traits are inherited independently of one another. It applies to traits whose genes are found on different pairs of homologous chromosomes. Remember, the homologous chromosome is one from mom, one from dad, to give you your pair of chromosomes. Your two, and on those chromosomes are the different pairs of alleles, okay? So, and during meiosis, genes for different traits are separated and distributed to the gametes independently of one another. So this kind of goes into play with the second law of segregation and recombination. So let's take a look. Let's look at an example here, okay? So here we have pigeons. You got a male pigeon with a crest, and these are his alleles. And you got a female who doesn't have a crest, and these are her alleles. So can these parents, the question on the bottom right, can these parents have males that have no crests? Can these parents have female babies with no crest? How many different combinations are there? Let's take a look. If you look at your notes, you will see that there's an area to fill in this box right here, okay? Now, of course, I'm not going to give you time to fill in all of these. You're going to have to go back and do these later, but each parent has 4 possible sex chromosomes. This is why that dihybrid cross that we did is going to come in handy, because that's essentially what this is. 4 possible sex chromosomes for each parent. The female, the mom, and the dad over here, okay? See? Dad, crested male. Possible gametes are Z and Z, so the sperm can only carry Z and Z, okay, for the crest. The mom, on the other hand, so sperm and egg, see? And then these would come down here, like that, okay. And then mom, Z and W, possible gametes, and Z and W, okay? So possible, and that's why they go here and here. Is everybody following along? I'm trying to go baby steps so everybody sees how this is created. And then once you have this created, you can start ZZ, ZZ, ZW, ZW, and you can get all of the sexes and the trait. Sex is also a trait, for the crestedness of each of the individuals, okay? So hopefully we'll have time later to finish that if you haven't finished that yet. Okay. Examples, some more examples in pea plants. I'm going to go back to the pea plants because they are good examples. This means the gene for seed color, yellow or green, is inherited independently from the gene for seed shape, round or wrinkled. Therefore, all combinations of these alleles for these traits are possible. The offspring could have seeds that are yellow and round, yellow and wrinkled, green and round, or green and wrinkled, you see. And this is actual picture of yellow round, green and wrinkled, so you can see that this was, real. In humans now, moving on from pigeons and peas, there's actually a pea called a pigeon pea, I think. That's funny. Okay, in humans, this means that the gene for earlobe attachment, free or attached, mine are, as you can see, free. Okay, they have a dip in them. Some people's are just attached, like right to the thing. I mean, this shows it right here. But so here you have attached, and it doesn't have too much of, and here's free. See how it curves up, and here it's attached? So it's inherited independently from the gene for freckles. Therefore, humans can have, humans can have free earlobes and freckles, free earlobes and no freckles, attached earlobes and freckles, or attached earlobes and no freckles. That's a weird word, freckle. When you say it over and over again, freckle, freckle, freckle. Now remember that this law only applies if the genes are on different pairs of homologous chromosomes. So you've got chromosome 1 and 2 from mom and dad, and then you've got other chromosomes over here, a different pair, and the gene for the other trait is on those chromosomes, okay? If the genes are located on the same pair of homologous chromosomes, they often tend to be inherited together, not independently. Can you think of any human traits that tend to go together? And we've been discussing a lot of traits over the past week plus, okay? But look around the classroom, think about your family, think about your friends, think about what you know about humans and traits, okay? So red hair and freckles. Oftentimes when someone has red hair, they have freckles. Blonde hair, blue eyes, right? Brown hair, brown eyes. You did know those. You like, oh yeah, I got it now. The tendency for groups of traits to be inherited together suggests they're located on the same chromosome, which is kind of what we just said. Each chromosome contains thousands of different genes. Thousands. Each of those chromosomes. Thousands of different genes on them. Because remember, you don't need just genes for how you look. You need genes to tell you how many fingers you have, and how many eyes you have, and how far apart your eyes are, and other things. And they're said to be linked because they're connected. Each chromosome contains thousands. Genes on the same chromosome are linked. So this is one chromosome. Looks like a propeller for an airplane, I know. But this is one chromosome, and if they're on the same chromosome, they're linked. Don't confuse this with what's coming up next time. Something called sex-linked, but it's sort of like that. You'll see in a minute. Those are chromosomes linked, or genes linked to the sex chromosomes, X and Y. So if genes are linked, they will not assort. That should be, will not assort independently of each other. So apparently Mendel was lucky to have chosen a trait in pea plants that actually does this on because it's located on different chromosomes. Had he chosen some other trait that was located on the same chromosome, he wouldn't have gotten the results he got, right? So how can genetic variation arise even when the genes are linked on the same chromosome? Why, for example, why don't all blondes have blue eyes? And I don't mean the fake blondes, the ones who dye their hair. Careful with that one, okay? So we know the answer. We know that it's crossing over. Because when during synapsis, during meiosis, when those genes would line up and those chromosomes line up and cross over, they actually switch. It's a really miraculous mechanism for creating diversity. Someone had a good plan. So we're going to watch a little video here. If I can get it to go.
So let's say you have to read an incredible non-email from your boss that you have to finish before the big meetings.
Human cells are diploid. We have received one copy of each chromosome from each of our two parents. When we make gametes, the sex cells, our gametes will possess one of these copies, but not both. But our chromosomes assort independently. Some of our gametes may, for example, have both maternal copies of chromosome 1 and 7, and others possess the paternal copies of chromosomes 1 and 7. But this is not the only possible pattern. Because the chromosomes are assorting independently, it is also possible for a gamete to receive the maternal copy of chromosome 1 and the paternal copy of chromosome 7, or the paternal copy of chromosome 1 and the maternal copy of chromosome 7. Our chromosomes assort independently. Chromosomes from one parent do not hang out in a group, always going to the same gametes.
I'm going to play that again.
Human cells are
I'm going to play that again, and the reason why is because I want you to hear it again, okay? And I want you to be clear on some of the terminology, because paternal, you may not be familiar with maternal and paternal. Paternal is dad, and maternal is mom. So when he's talking about maternal and paternal, please know that those are the mom and the dad, okay, the parents. Paternal, P, dad, and maternal, M, mom, okay? Let's watch it one more time. It's only a minute long. So listen very carefully. He shows two scenarios of the same process, but you can see that the chromosomes assort independently. They assort a different way both times. It's pretty cool.
diploid. We have received one copy of each chromosome from each of our two parents. When we make gametes, the sex cells, our gametes will possess one of these copies, but not both. But our chromosomes assort independently. Some of our gametes may, for example, have both maternal copies of chromosome 1 and 7, and others possess the paternal copies of chromosomes 1.
So the paternal is the blue, and the maternal is the red, right? We got that? Okay.
And 7.
Or
But
pink.
this is not the only possible pattern. Because the chromosomes are assorting independently, it is also possible for a gamete to receive the maternal copy of chromosome 1 and the paternal copy of chromosome 7.
You see, so now you've got blue and pink on each side rather than blue on one side and pink on the other.
Or the paternal copy of chromosome 1 and the maternal copy of chromosome 7. Our chromosomes assort independently. Chromosomes from one parent do not hang out in a group, always going to the same gametes.
All right. So that is independent assortment. So again, going back to crossing over, it occurs when segments of homologous chromosomes are switched, right? During meiosis. So they line up during synapsis, the chromatids cross, and then they trade. See, first it's BB, big B, big B, little b, they trade them off. Crossing over happens. And there are many genes on here, thousands of genes on the chromosome. And so you get new genetic combinations. Again, that is why you don't look like an exact copy of your mom and dad. You may not even look like a perfect mix of your mom and dad because of this. So in the diagram below, what are the original linkage groups of alleles? The alleles are A, capital B, or little a, little b. See? But in the diagram, what are the new linkage groups, combinations of alleles that have formed? you've got A, B, the recombinants, or a little capital A, little b, or a little a, capital B, right? So crossing over increases the genetic variation. And I told you why. I'm going to talk more about this in the next unit, which is evolution, about survival. This may cause, this is essentially a genetic mutation, but it's not a bad thing necessarily. Mutations aren't bad things. Mutations are just changes in the genetic code. And if, let's say, a new virus comes, like a new corona type of virus, some people will have a natural immunity because that their parents don't have because of this, okay? And now we're going to talk about multiple alleles. Multiple alleles, in all examples given so far, there have only been 2 alleles for each trait, right? Big H, little h, big H, big B, little b, big S, little s, okay? So yellow, green seeds, red or color white, attached earlobes, free earlobes. But in some cases, there are multiple alleles. That is why we have not just blue and brown eye color. That's why you don't just have short people and tall people. You've got lots of things in between, right? So that's because more than one pair of alleles codes for that trait. So for such traits, it's important to understand that although more than 2 alleles exist in a population, each individual organism can still only have 2 of the alleles, one from each parent. But there are multiple ones out there in the population. So let's talk about human blood types, because if you look around the classroom, we're going to have people who fit into all of these blood types. There isn't just 2 blood types. There isn't just A and B, or let's discuss it, okay? So when you symbolize, all capital I is dominant and lowercase I is recessive, okay? One recessive, but there's more than one dominant. That's where it gets tricky, okay? So human blood types are all the result of certain proteins embedded on the surface of the cell, in the cell membrane. And there are 3 alleles that code for these proteins. And they're here they're on your notes, okay? So I think I left this, I think you have to fill that in on your notes. Yeah. So the 4 different phenotypes and the 4 different genotypes that go along. Please make sure you get these superscripts, super above, script, writing, superscripts, properly. Don't put lowercase a, make sure that they're capitals. Do it exactly as you see here on the screen, because you're going to need that information to create a Punnett square for these, to see who the offspring will be, what the offspring's blood types will be. And again, these are all based upon, different people's blood type is all based upon the proteins that are embedded in their cell membranes of their red blood cells. And your body is genetically coded in the DNA to produce red blood cells with certain proteins. And if you got blood from somebody else, it could either be fine or it could kill you. We'll talk about that in a minute. So you should at this time have had enough time to copy all of that down. If not, you're going to have to come back to it later, okay? All right, so the alleles for blood type are I, capital A, I, capital B, and lowercase I, okay? The recessive one. No absence, the absence of cell proteins, okay? should have those 3 letters down. We're on slide 71, so we have only 6 slides. We're going to stop when we talking about blood type. So we have a couple Punnett squares to do on the next page, but not too bad. So the alleles for red blood, A and B, are codominant. We talked about codominance in the last set of notes when we talked about horse color and cow color, remember? This means both alleles are expressed. The horse is both brown and white. It's not a mix of brown and white. Tan, it's not like that. The proteins embedded in the membranes are called antigens. So you can see antigen A, antigen B. There's something called Rh factor, which I don't think we cover in this class. Just another protein. That's why when you get married, you have to get a blood test to make sure that you and your spouse have compatible blood types, because your baby could have a chance of having something wrong with their blood. In addition to red blood cells, human plasma also contains proteins called antibodies. And this is what I was talking about before if you got the wrong blood type from somebody. Because the wrong blood type would be a foreign cell in your body, and your body will attack it. When antibodies encounter a foreign antigen, like the markers on the wrong blood type, they combine with it, like this, antibodies, and they stick them together, they clump them together. Quite serious and often fatal. The person most likely will die. If you hook up the wrong kind of blood, and apparently right now, I just read in the news, there's a national emergency blood shortage. People, because of COVID, people are not donating blood. They're not going out and donating blood. So if you get in an accident and you go to the hospital, they might not have your blood type that you need. Maybe they'll have 1 pint of it, but not 6 pints in case you needed a major surgery or something like that. So if you are of age or approaching, I think, 16, you can, with parent permission, you can donate blood. You have to be over 100 pounds, I think. All right, so this big table is on the bottom of page 15 for you guys on your notes. And you can see the genotypes on the left. And you can see that some of them are going to give you the same kind of blood, okay? And then some of them are standalones, all right? So the first one is going to be type A. That's kind of obvious because the A's are there alone, okay? That one's recessive, so this one's going to be dominant over that one, okay? Type A, same proteins. Type B is going to be virtually identical to A, except replace the A's with the B's. And that's supposed to be a capital B. Is it a capital B in your notes? No, fix it. Capital B. Typo. If you have both alleles, one from mom, one from dad, A and B, you are type AB blood. And then if you have neither A nor B and just the 2 recessive alleles, you're type O blood. And you can see type O is the universal acceptor. You can get anybody's blood, A or B, and your blood won't clump. Type A can only get type A. Although if it gets type, if it gets type B, it will clump. If this, and same here, reverse, opposite. Type AB can only accept type AB. If it gets either one, it will clump. So and then this part is on the bottom of page 15. Most common are O positive and O negative, and that has to do with the proteins, the negative part. The most rare is AB negative. The universal donor is O negative, and the universal acceptor is AB positive. Five seconds, come on, get it down. Okay, so here we go. Human blood Punnett square. All right. So this one I'm going to do with you. I'm going to guide you through this, okay? So these are our parents. Let's imagine that our parents are type A, heterozygous, they have a recessive allele, and type B, see that's supposed to be a capital B, not a lowercase b. There's no lowercase b's. Heterozygous, they have the one recessive allele, so they are going to be type B blood. This parent is going to be type A blood. So let's finish the Punnett square. It's just putting this and this in there, right? So you're going to be AB. 1/4 of the offspring will be type AB blood. 1/4 of the offspring will be type B, just like that parent. 1/4 will be type A, just like that heterozygous, just like that parent. And 1/4 of the children, probability, right, the chances of having a child will be type O blood. So now you can definitely do the genotypes over here. So that's going to be 0, 25%, 0, 25%, 25%, 25%. And then the phenotypes, 25% type A, that's the bottom left. 25% type B, that's the top right. 25% AB, top left. And 25% type O, bottom right. It's actually pretty easy. Those little symbols, the little superscripts, you think, oh, that's going to be scary. It's not really. It's kind of easy, right? Okay, so here's an example. It's in your notes. A woman who is heterozygous for blood type B marries a man who is heterozygous for blood type A. Using a Punnett square to determine the possible, blood types of their children. Remember, step A was to have a key, so that key will typically be given to you. And then step 2 is to find out which gametes are possible by each parent. And so we just essentially did this on the last slide, right? But you're going to fill these in the blank spaces. And then you've got capital IB, little I, capital IA, little I. So these are the possible gametes. I think we're kind of going backwards here, but yeah, we did go backwards. Ha ha ha. Okay, that's fine. But example 2 is forwards. So Sheila and Tom are married. Right in your notes. You can read along in your notes. Sheila, who is heterozygous blood type B, has a daughter, Katie, who is heterozygous blood type A. When Katie developed a serious genetic illness, it became critical that her complete hereditary background be determined. So 3 other men came forward, all claiming to be Katie's biological father. This class just switched over to like Maury Povich or something like that, TV show, or Jerry Springer, right? Okay, who is the dad, right? And the DNA test comes out, okay? So, just kind of flashed the answer there, but you have to use a Punnett square to prove your answer. So on your sheet, I want you to use your use the information. You're going to do it. I'm going to give you the answer, but you have to prove it in your notes. Okay? So here, the answer is Joe the milkman, type A, B. So show me why it's that in that Punnett square. Does that make sense? Okay. And you're going to do that later, not now. And that's our notes for today. So you do have time in class to go back and finish that other part of the notes that we just did and this one. Okay? And we'll do part 4 next time.